start. So, welcome back to reality. Um, Unfortunately, it is still chapter 10. It's only 10.3. You see how slow this thing goes? Right. Okay, so just a quick recap from the previous two lessons in 10.1 and 10.2. We dealt with the carbon reactions, but the specific part of the carbon reaction, namely you learned about the Kelvin cycle itself, the three phases of the Kelvin cycle, and all the enzymes that involved in the Kelvin cycle, and also the concept of formation of sugar precursor, the glycerol dehyde 3-phosphate, and why the process needs to take place in the form of cycle rather than a linear biochemical reaction, okay, because of the regeneration phase of the cycle itself. And this is all true for the regular C3 plants, okay, which happens to be for the most terrestrial plants on the planet. But C3 plant is not perfect. It's got its own evolutionary issue. It's very ancient, and the issue there is the photorespiration. Depending on your context, if you are talking about the performance of the plants, formation of sugar, and so on, you might want to regard that as a wasteful biological reaction, the photorespiration. Remember, photorespiration, it's not only happening in the chloroplast, that actually took place in three different organelles. What are the organelles? Chloroplast, peroxisome, mitochondria, and then come back to chloroplast. Okay, so the whole idea of photorespiration is to salvage the CO2 back into the system while removing the 2-phosphoglycolate, the C2 carbon compound that happens to be toxic. So the plants cannot afford to have this lingering around and about for too long in the vicinity. But because there is no specific set of enzymes to deal with this breaking down of phosphoglycolate, that's why it involves the peroxisome and mitochondria to achieve the goal. But of course, at the end of the photorespiration lesson, you see that some people are actually working on this, because some bacteria, they actually can perform this carbon concentrating mechanism that can bypass all these needs to salvage the carbon back into the system. Okay, so let's move on for a bit. So you have learned about the C3, C2, C1. That I want to ask, what is C1? Somewhere. Where do you find C1? C1 metabolism. Hint is in the photorespiration story. Somewhere. Please revise that, okay? So today we're going to look at the next carbon metabolism. It's called C4. So this from now on until another two lessons, you're going to see that the concept of carbon concentrating mechanism. The idea is simple. This is all to deal with the inefficiency of Rubisco. You see Rubisco? It's an ancient enzyme. It's got dual affinity. It can catalyze two biochemical reactions, the oxygenation reaction and carboxylation reaction. If it does carboxylation, then that's good. You got your sugar precursor and so on. But if it does the oxygenation, then you come with this wasteful, metabolically wasteful photorespiration. Why is it off? That thing keeps doing it. Is it really off? Tekan balik. Maybe they put timer. Nobody wants to pay the bill. Right. Yeah. So we move back. Tak apalah, it's okay. Right. Okay. So there are two more carbon metabolism that employs this carbon concentrating mechanism that you're going to see today. Okay, the C4 and also the CM. Oh, suddenly quiz, right? This is actually a pretest. Not to worry, we're not going to do the quiz now. We do it in the afternoon. Right. So let's dive in. What's the story about? So, are you familiar with evolution? Did you learn evolution in any of the classes before? Pernah belajar evolution before? Not heard. Some crazy old man. Why? Missing notes? Have you learned evolution before? Learn it as in learn it. Did you take your soil science? Did you learn the Bowen's reaction series? Do you know what is Bowen's reaction series? You got your lava is solid, and then it solidifies. The first thing you get is the glassy rock, the obsidian, the pumice, and so on. Did you learn that? Oh God, people. So in soil science, you learn there is a section about the rock genesis. Okay, so that's where you learn about the metamorphic rock. You learn about the igneous rock. You learn about the sedimentary rock. These things all take a very long time to happen, right? Especially the sedimentary rock and also the metamorphic, right? Igneous rock is kind of quite straightforward. From the lava, and then it cools down, then you get your rock. Igneous, Latin, it means fire. Okay, so I use this example because you learn about that, right? The concept here is kind of similar like the evolution. With the progression of times, the things changes. Just the effect of time to it, because time makes a lot of other events happening on the subject. In the case of metamorphosis, what is the factors that change the original igneous rock? You start with the igneous rock, and then after 200 million years old, it changed into a metamorphic rock. So what are the factors affecting on this? Pressure, temperature, cementation, macam-macam. So many things. Then you got your metamorphic rock. So from the slate, you get your marble. From sheets, you got quartz and so on. Macam lah. Right? I should be teaching soil science, right? Right. Okay. So evolution, kind of like that. Yeah. So the terminologies of evolution is not only restricted in the case of from rat, you get human, you get orangutan. Okay, it's the concept of the change of developmental physical of something, right? The baby, the embryo, the zygote in the fetus evolved to form a full-grown baby. It started with just 2 cells, right? So it will evolve. So the concept also applies here. You see, most of the plants actually C3 plants started. That's the ground of it. However, due to the decline of oxygen. Not the decline of oxygen. The decline of CO2, the increase of oxygen and so on. It's causing the function of Rubisco, which started very long time ago, not anymore suitable for the current environment. Meaning that Rubisco, it's actually quite happy with whatever environment it was from 200 million years ago, right? But now, what has changed now in terms of oxygen, carbon dioxide concentration? In the past, do we have more CO2 or less CO2? Less. Okay. Why? Because at first the photosynthesis is still not around. You still get all these individual bacteria and microbes that still do not do this endosymbiotic line or activity. Okay, so they are pretty much producing CO2. Okay, because there's no photosynthesis. But with the advent of photosynthesis, there is a booming of oxygen. Boom. Which is good because other life started to appear. And this is why you get all the dinosaur and birds and so on, including your sinful ancestor. The problem is Rubisco is not equipped with this. Okay? It's kind of happy in the high CO2, but not in high oxygen. However, it still needs to do the job, but it doesn't really evolve until now. So some plants, they had to go through with this, and on top of it, certain plants, especially in the hot, arid climate, think of Brazil, think of Peru, think of Mexico, think of certain dry area in Thailand. They got the distinct dry period, right? So the plants in this region, they have actually undergone very difficult situation in addition to the inefficient Rubisco. They have to deal with the Rubisco stubbornness to do photorespiration. And then they need to reproduce. So over the time, the plants actually have started to evolve their self-characteristics. Two important things. They change the biophysical appearance of it, meaning that they change the leaf structure, they evolve, and also they change the biochemical pathway inside the leaf. Right? It is still plant. Okay, it's not like suddenly it evolved to become some kind of alien. No. If you go out and you compare between the corn and other plants, grass plant, it still looks like a plant. Okay, so suddenly the corn is wearing a skirt. It looks similar on the outside, but anatomically it's different. Biochemically it's different. So what's the difference? So that difference is very crucial to deal with photorespiration. Remember, the whole idea is to prevent photorespiration from happening, right? So for the C4 plant, the reason it is called C4 is because the first carbon fixation event, the first product of it, is not phosphoglycerate like you learn in Kelvin cycle. Okay, but rather the first product of it is a 4-carbon compound. Okay, such as malate here, right? So the image that you're looking at here is a typical representation of a C4 photosynthesis organism, such as corn, maize. You can see there is a first cell here, mesophyll cell, and bundle sheath cell. This is rather different because previously when you only learn about mesophyll cell only. And suddenly there is another cell now. Yeah, they got a function. So let's go through this. CO2 in C4 plant gets into the mesophyll cell. And remember, okay, mesophyll cell is actually filled with water just like any other cell. And CO2 can dissolve in water. The moment CO2 dissolves in water, what do you get? You get carbonic acid, right? And this carbonic acid actually can further be dissociated into bicarbonate ions. Okay, and this is very crucial because this bicarbonate ion is the one that is going to react with a special 3-carbon compound called phosphoenol pyruvate. This phosphoenol pyruvate is not a sugar precursor, it's just a 3-carbon compound that going to capture the CO2 in the form of bicarbonate ion to form malate. 3 carbon here, 1 carbon here, you got 4 carbon. This is why it is called the C4 metabolism, right? So this is all achieved with the presence of number 1, carbonic anhydrase, and also PEPCK, phosphoenol pyruvate carboxylase. Carboxylase means the incorporation of carbon into a compound. Decarboxylation means the removal of CO2 out of the compound. So the terminologies are very important here. The one thing about the carbonic anhydrase is it can do this thing. I don't think it's in the book. Something like this. How does it start? So you got your CO2. Remember, this is all liquid, okay? Not gas. CO2 plus H2O. Then it goes kind of like, should it go here? Should 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 go here? It means reversible reaction, okay? Carbonic anhydrase. So this you will get H2CO3. So this is carbonic acid. So this thing can be dissociated into bicarbonate ion H2, no, not H2, HCO3 minus plus hydrogen proton. Okay, so you can see that carbonic anhydrase can make CO2 that has dissolved in water to become carbonic acid, and then from carbonic acid it can further change itself into bicarbonate ion. This is bicarbonate ion. You know your bicarbonate that you use to make whey? It's the same thing. Bicarbonate plus hydrogen, and from this it can revert back to CO2 and water. So it's kind of like this triangular reaction with carbonate anhydrase. Yeah. So the star that we are talking about here is this guy here. This is the important thing. Bicarbonate ion. So this bicarbonate ion now reacts with phosphoenol pyruvate, or short is PEP. This PEP now becomes oxaloacetate after it has reacted with bicarbonate ion. Look at here, okay? It becomes oxaloacetate plus PI, inorganic phosphate. Sometimes in the book it doesn't say PI, it says orthophosphate. Kind of the same thing. Okay? Right. Now, look at here. You are still in the mesophyll cell, okay? We are not in the bundle sheath. We're still in the mesophyll cell. This oxaloacetate, two things can happen depending on the subspecies or subtype of the C4. Some species from the oxaloacetate, it will be reduced, you get malate, or the oxaloacetate react with an amino acid undergoing transamination to get another kind of amino acid called aspartate, class 2 oxoglutarate. If you recall from the photorespiration lesson, you found this guy a lot, 2-oxoglutarate. The moment enzyme aminotransferase is present, always, all the time. Kau tak percaya, kau tengok. Saya ada benda tu. When you found something is using enzyme aminotransferase, this guy 2-oxoglutarate will be present. So this is the reduction process. Reaction number 3 is the reduction of oxaloacetate. Number 4 is the transamination reaction. In any case, you get 4 carbon compounds. Either malate, if the plant is using reaction number 3, or you get aspartate if the plant is using reaction number 4. Okay? I'm sorry, but memang kena tahu these two because some plants, they kind of overlap, they ikut mood dia. Right? That macam, because the evolution is actually still not finished, betul-betul. So there is an overlap and some plant kind of do halfway, lepas tu follow the regular C3. Right? Okay. So, clear with that? Yeah. So, you have finished the first part of the C4 step, which is carbon fixation. But this is in the mesophyll cell. So, from the mesophyll cell, the 4 carbon compound, either malate or aspartate, will move to the second cell, which is called the bundle sheath cell. Sheath meaning the cells is actually in the concentric arrangement surrounding the vein. So if I may draw the vascular system like this, so in the middle here, you got your vascular system, xylem and phloem. Surrounding here, you got your bundle sheath. Outside the bundle sheath, you got your mesophyll. So the key point here is bundle sheath is right next to the vascular bundle. Because the Calvin cycle that you are familiar with is happening in the bundle sheath cell, not in mesophyll cell. Remember, okay, you're not dealing with C3 now, this is C4. So the situation is special. It's a bit unusual. Okay, so what happened in the bundle sheath cell? So when the original C4 compound, malate or aspartate, has entered the bundle sheath cell, it needs to undergo decarboxylation event. So this 4 carbon compound, the CO2 in its body needs to be taken out. Why? Very easy, so that the CO2 can proceed with the regular Calvin cycle. You still have your Calvin cycle, but it is now specialized in the bundle sheath cell. So malate here will release its CO2, and then this CO2 proceed in the regular Calvin cycle. And this is the critical step here, because malate here will incessantly release CO2 to jam Rubisco with CO2 carboxylation mindset. There is no room for oxygen for you. So that's the whole idea. Use a compound, ask the compound to go in crowd towards Rubisco, and all these compounds bombard Rubisco with CO2. So rubisco has no reason to do oxygenation at all. In fact, the efficiency of this thing is photorespiration, if it ever going to happen, only about 2% to 3% only. That's how good this carbon concentrated mechanism is, right? You can see the enzyme here, NADP-ME enzyme, the enzyme that dissociate the 4 carbon compound into carbon dioxide and pyruvate. Pyruvate is the 3 carbon compound that can be shuttled back to the mesophyll cell. CO2 is released to proceed with the Kelvin cycle. And then at the end of this, you get your pyruvate, the C3. This is not a carbon or sugar precursor. No, this is just a 3 carbon compound. This 3 carbon compound pyruvate, or in some species, there's a section here, alanine. Depends on the species. Pyruvate or alanine go back to the mesophyll cell so that it can be converted to phosphoenol pyruvate, PEP. And this PEP can redo the 4 carbon fixation again, and the cycle continues. All right. So pay attention to this enzyme here. There are 3 enzymes here: NADP-ME enzyme, NAD-ME enzyme, and phosphoenol pyruvate carboxykinase. These 3 enzymes actually also the name of 3 variants of C4 plants. You see, C4 plant got variants as well, depending on the species. I'll show you in a bit. Depending on the species, it will decide which enzyme to use, whether it is a NAD-ME, NADP-ME, or phosphoenol pyruvate carboxykinase. Right? Okay. Yes. 3 variants. Yeah, 3 variants. What about corn? Corn, sorghum, it uses the first one, the 5A, NADP-ME enzyme. For the millet, you know millet? Millet use the 5B, NAD-ME enzyme. Some grass, like the Guinea grass, you know, people use Guinea grass for the fodder pasture. That uses number 6, phosphoenol pyruvate carboxykinase. So this enzyme, despite all these hateful different names, the function only one: decarboxylation, meaning that the moment it is going to do its work, it will remove CO2 from the compound. That's all its job, despite all these crazy names. Okay, right. So now, at the end of this reaction, you get your pyruvate or alanine. Again, this depends on the species as well. It will be transported back to the mesophyll cell, recycle into phosphoenol pyruvate, and then come back again to do the C4 carbon fixation. Right. Okay, so basically this is just to show you whatever I have said verbally. So this is the story here. Recap. You are using 3 or 2 different locations here in the cell. Never ever say carbon assimilation, carbon Kelvin cycle happening in mesophyll cell. No, wrong. Don't say that. Your regular C3 happening in bundle sheath cell. Okay, and bundle sheath cell is right next to the vein. So that whatever sugar that is accumulated here, because the rule is still similar here. When a chloroplast is jammed with sugar, it cannot make new sugar. This sugar has to exit first. What is the best way to exit other than sitting next to the piping system? Right. So next to the pipe, you got lots of sugar, just unload the sugar. Now you can make more sugar. So this is why C4 plants such as corn super fast. because it is not sleeping at noontime like C3 plant. It can do photosynthesis at all the time. There is no midday depression. There is no need. If the stomata is closed, then that's fine as well. You can get the harvest for corn in 70 days. From seed to seed, you can get it in 70 days. But for rice, from seed to seed, 2 times the length, 140 days. So this is why Bill Gates and many other teams in the world is looking very keenly to change C3 into C4 plants. But of course, this is not easy. You can see, you need to worry about the structure first. If the structure is incorrect, you're not going to get it. However, they seem to have found a little light at the end, because in certain plants, the C4 metabolism do not happen in separate cells, but rather happening within the same cell, like the plant here, Bioinertia and Sueda here. So these two plants here, they are C4 plants, but they do not have this concept of mesophyll cell and bundle sheath cell. They just have the photosynthetic chlorenchyma cells. However, the fixation of the carbon happening at one end of the cell, which is close to the atmosphere, and then the assimilation, the regular Calvin cycle, happening in the middle of the cell. So we call this carbon compartmentalization. Yeah. So the job of fixation of carbon close to the atmosphere, the job of carbon assimilation happening over here. So when this happens, okay, you can like it here, the morphology of the chloroplast is not similar. Okay, you got we call it dimorphic chloroplast, because chloroplasts in this region usually they do not have photosystem 2 active in the assimilation region, because if the photosystem 2 is active, you get your oxygen, right? Here you don't want oxygen, because the moment you get oxygen, you get photorespiration. So in this chloroplast that is doing this C4 metabolism, photosystem 2 is lacking. They just have the Calvin cycle. Yeah, right. But for the mesophyll cell, the regular mesophyll cell, you can find from time to time photosystem 2 and 1 active. Yeah, but not in the bundle sheath cell, or for the case of one cell C4 metabolism in the carbon assimilation region. So this region here, no photosystem 2, because you don't want oxygen. Why? Photolysis in the carbon in the light reaction produce oxygen from water splitting. You don't want that. Okay. Right. Okay. That's the regular depiction of it. Yeah, you can see there is no distinction of the cells. Just you've got your stomata, CO2 gas in, and then all happening in one go for the C3 plant. For the C4 plant, mesophyll cell closest to the atmosphere, CO2 gets in here, CO2 becomes the C4 compound, C4 compound gets into the bundle sheath, and then it gets decarboxylated, and then the regular Calvin cycle can happen, and then the C3 compound come back to the mesophyll, so that C4 compound can be formed again. Right. Oh, I'll put an example here. So these are the three variants of C4 subtypes, we call it. Okay, depending on what? Depending on the decarboxylating enzyme. So you have type 1, type 2, type 3. Most of the time in all textbook, you only learn this thing. But because this is a Cambridge Oxford bound class, you get to learn the other two. So the first one, the regular one, is this. It's very easy. You fix your carbon CO2 using the HCO3. What is HCO3? bicarbonate to get oxaloacetate. Then oxaloacetate will be converted to malate. Malate go to bundle sheath and then get decarboxylated. CO2 proceed with Calvin cycle. The leftover is pyruvate. Pyruvate come back to become phosphoenolpyruvate in the mesophyll cell. For the second type, you see there's a difference here. It's no longer malate. It's aspartate. Don't worry, it's still 4-carbon compound. It's just a different arrangement of molecule. Now it becomes aspartate. Still a type of amino acid. Okay. Aspartate go to the bundle sheath, but it will be converted to oxaloacetate again before we come back to malate. So this is a bit longer, right? Come back to pyruvate and then becomes alanine. Alanine change back to pyruvate, then become the phosphoenolpyruvate. For the third type, the third type is even longer. Why? This is the question why. This is one of the proof this thing is due to evolution. Because some plants have a quicker way, someone have the shorter way, some plants kind of confused, have both ways. It looks uncertain. The biochemical pathway looks uncertain, even though the plants belonging to the same family. So, evolution as we know it, when it's still ongoing, nothing is fixed, anything is possible, right? So this is the example to show you how evolution is still take place. Maybe this is the latest, the most advanced evolution of C4. Maybe this is the start of it. Maybe this is 100 million years ago. This has been around 150 million years ago. This has been around since 200 million years ago. So this is the longest. So that's why it looks the simplest. Right? Okay. Why? Why it can become simpler, but this one no. If you think about it, compared to corn and guinea grass and millet, who are more heavily used by human? Corn. So when humans interfere a lot, the speed of breeding becomes faster as well. When that happens, this evolution is cut by so much time. Right? The domestication of it. The domestication not only to increase the yield, other things can happen as well, including biochemical improvement. This is biochemical improvement. Right? So in addition to the hot climate and so on, acting upon the plant and the plant have to do with it, the farmers can be blamed for the evolution as well. Because they look for the plant that can withstand the Mexico super hot, unpleasant situation. Right? So this plus nature, you get the most advanced of it. Maybe one day, don't be surprised, corn become this as well. No need for 2 cells, just 1 cell. But this, for now, this is kind of difficult. This is actually for the much simpler plant, right? Actually, this is more efficient because in here, there is no oxygen. In here, maybe oxygen, you still get oxygen here, right? And the oxygen can diffuse because this is still within the same cell, right? Okay. Oh, what's that? All right. So whatever that I just rambled about since 10 minutes ago, this is the story of it. And you should know the start of this class. I ramble first, then you read later. Right. So that's the essence of it. The hydration of CO2. This thing is this thing. I noticed in the book it do not have it. Did you learn this in your biochem class? You should learn this. This thing happening in your body as well. Your body, blood, got CO2, right? The CO2 needs to be released back so that you can exhale out, right? Carbonate anhydrous. Right. Okay. So, yeah. Number 5. That's a bit important. Number 4 and 5. We're talking about the diffusion back to mesophyll cell after done with the decarboxylation. The 3 carbon backbone that come back to the mesophyll cell in the form of pyruvate or alanine. So this thing depends on the species as well. However, the nomenclature based on the enzyme. Right? Okay, look at number 5. The regeneration of phosphoinopyruvate and also the bicarbonate ion, which is the acceptor, consume ATP. Now we need to know this. ATP. In order to do all these fancy schmancy magic tricks, ATP is needed. So for every one CO2 molecule fixed, you need about 2 ATP. So we call in C4 metabolism, this is some kind of like a metabolic investment. But the plants, it's okay. You invest 2, but you get back 20. Who cares? Right? Rather than you have to go through the wasteful photorespiratory path. Right? Does it say in here? 2. I think it should say somewhere. Here. 2 molecules of ATPs are consumed in the conversion of pyruvate to phosphoinopyruvate. 1 in the reaction catalyzed by pyruvate phosphate dikinase, reaction number 8, this thing here, reaction number 8. So you use 1 ATP there, and another in the formation of AMP, adenosine monophosphate, by adenylate kinase. So that will be reaction number 9. So 2 times ATP is used. So that's why it says 2 ATP per CO2 molecule. Right, okay. This is the summary, what I just said again. The type of plant, C4 subtypes, and the enzymes that it uses for decarboxylation. For the C4 transport, this use malate. For the coming back from bundle sheath to mesophyll, it uses pyruvate. Okay, you got your sorghum, you got zimase, then you got cleome, triplex. Cleome, we got the plants here. It's kind of like weeds. Triplex is, what's that? This is common in Middle East, pokok ni. Triplex is a, what's the name? Salt bush. Do you eat bayam? Triplex is the same family as bayam. Amaranth. Yeah. But people in Arab don't turn it into masak lemak lah. And then Megateras, I think this is the new name for the Guinea grass. Guinea grass that I learned from long time ago, nama dia Panicum maximum kot. That was from 20 years ago. I think the name has changed now. You can double check. I think this is nama baru Guinea grass. Right. Okay? Right. Alright, so this is also the summary what I have just said. So it's very important to understand the concept of decarboxylation depends on the species, the enzyme that it uses, and also the forms of the chloroplast. Okay, the chloroplast is not randomly scattered about. I think it should say in here somewhere. Let me see. Is it here somewhere? Oh, wait. I don't know whether what it does say, but I just know the knowledge of it. You see, this NADP malic enzyme, this actually happens in chloroplast. This NAD malic enzyme, this happens in mitochondria. We're talking about bundle sheath, okay? It's still in the bundle sheath cell. This phosphoinopyruvate carboxykinase, this happen in cytosol. You can read. If the knowledge is there, then adalah. I do not know whether it's mentioned in here or not. Ada ke? Ada ke? Oh, different slide. I got it wrong. But the info is there. Okay? Right. Okay. So, what about the structure just now that you saw? That structure got a name. It's called Karanz anatomy. So this is the cross section of the rice leaf. This is cross section. You see here, the one in the middle here, this is the vascular bundle, the vein. The concentric circle cell here that you see, swell up here, this is the bundle sheath cell. And the green part here, this is the mesophyll. The green part here, there's another name that you can call it. Mesophyll, when there's a lot of mesophyll, we can call it chlorine cell. Remember you learned parenchyma, chlorine. Yeah, parenchyma that have photosynthesis activity we call chlorine. They got the name from chlorophyll. So meaning that chlorine, the green tissue, does it look green? So this is all chlorine tissue. Regular C3. The B is the C4. Look at here. The super concentrated deep green happening where? In the bundle sheath, right? So this is the Karanz tissue, so to speak, right? So what about the rest of it? The rest of it just to fix the CO2, right? Fix the CO2 and then shuttle to the bundle sheath cell, and then bundle sheath cell, once the sugar has been formed, go straight into the vascular bundle. This is the cross section of it. But what happens when you look at the physical of the leaf? This is something that we can do in the lab later. When you look at on the leaf, this is how it looks like. For the Karanz anatomy, you will have higher vein density. You see, from the middle of the vein for the Karanz anatomy, you only need about 2 cells. 1 cell, 2 cells. But in between C3 vein, from this vein to vein here, how many cells? There's so many cells here actually. It can be as high as 15 cells, right? So when this happens, you get this on the leaf surface. Okay, you get less vein for the same distance. But when you have Karanz anatomy, you will get high veins with the same distance. So which one is good? Less plumbing system, more plumbing system. Why? Yeah, you can transport the sugar source to sink very fast. No need to wait. Sugar accumulate for long to do what? This is not storage organ. This is the source organ. So it needs to transport very, very fast. So more piping system, that's good. So one of the things that they try to find in rice is actually, if they don't manage to turn it into C4 plant, find rice or a treatment that can increase venation. Increase the venation. Not only that, it's good for the sugar exit. It will cause the leaf to be strong as well, because the vein is actually kind of like a skeleton, right? You know, rice is easy to lodging. If you have more skeleton, it gets stronger. Right. Okay. This is all the story here. Benda aku dah cakap. Ni benda ni. 2 to 3%. It get reduced. Sometimes it's zero actually. With this compartmentalization of bundle sheath and mesophyll cell. Aku tak puas hati. Benda sheath macam mana? Ini. Ada kat sini. Ni. NADP ni happens in chloroplast. NAD malic enzyme happens in mitochondria. Phosphoinopyruvate carboxykinase happens in cytosol. At least kau tahu aku tak scam. It's in here. I read it long time ago. I just know dalam buku ni disebabkan it's the latest. It should mention somewhere. Tapi tak sure mana satu. Inilah benda ni. Right. Okay. Right. Lagi 2 slide lagi. Then we can be done. What about the single cell just now? So the single cell just now sebenarnya dia happen kat mana? If it's not in terrestrial plant, it actually happens in the aquatic plant. So in the aquatic plant, you got your seawater and everything, right? So this actually, with the help of Ca here is the carbonate anhydrase. This guy here, carbonate anhydrase. After the water has been filtered, kena filter lah. This is seawater. Kau nak kena darah tinggi. It filters and it gets the bicarbonate, and this will be sent straight to the rubisco site. But for this to happen, this is actually happening. One cell. So this is seagrass. I think this is my macro algae. Yeah. Remember, it doesn't have to be green. Sometimes this algae warna merah. If you remember from the previous lesson, the lineage of the photosynthesis, some go to green, some go to red, some go to kelabu, right? So this is some of this actually red, right? Red doesn't mean that chloroplast is not present, okay? It's present, but the color is not dominant sometimes. Okay, so what about it? So is light important? Yes, light important because light is actually responsible to activate many of the enzymes that you saw on this table here, table B here. For example, NADP-ME dehydrogenase is dependent on ferredoxin. You see this guy again. Remember you see this thing before? You saw this thing before when you were learning about rubisco activation. You see this again. Who else is dependent on this? NADP-ME dehydrogenase, which is in reaction number 3, I think. Check number 3. Yep, number 3. Meaning that without light, dehydrogenase of malate is not activated. If it's not activated, how on earth is it going to capture CO2? It's not going to happen. And some of the unusual things also happening because at night, some of this enzyme needs to be down-regulated. Which enzyme? Let me see. Yeah, this phosphorylation of this in the darkness, it needs to be activated because in the darkness, well, there's no photosynthesis, right? So why waste the metabolic resources? So plants rely on the activation of this compound to up-regulate or down-regulate this enzyme so that they are doing the job in the right manner, right? If they are active during the night, when there is no ATP produced, because remember, these two cells, they kind of not having photosystem 2 in both of them. Only one of them got it, right? So they need to have some kind of communication to tell we are ready because we have been activated by the sunlight. So now you can enter. The other side needs to tell we can't do much now because the darkness has struck. Some of our guy has been deactivated. So no more resources for you. So stop doing stuff. Right. So this communication happening at on regular basis for the purpose of what? Easy. If the plant is active all the time, when is it going to grow? Plants, dia tak tidur. It's not sleeping, but it needs the darkness to do elongation, to relax the cell wall so that the expansion can take place. Yeah. Not to say that in the day, expansion is not happening, but when it's darkness, the starch can break down. Starch yang buat pagi tadi, it can break down. Then this can be used as energy for the growth and expansion until the light comes back in. Yeah. So if you think about it, that is smart. It means the plant know how to utilize the time at the best of its capacity. During the day, when the energy is available, stop enlarging, stop growing, but acquire all the energy that is possible. At night, when there is no light, there is no energy, break down the energy, do the expansion, do the elongation. Kalau dia buat dua-dua waktu pagi, kan tak efficient. If it's growing during the daytime, who's going to do all this biochemical pathway? Right. So nampak tak? Dia ada pembahagian kerja. So that's the smart thing about this whole thing. Okay. Right. So this is pretty much aku dah cerita semua benda ni. Yeah. Yep. I've mentioned it. This is about the apa tu? The movement of the pyruvate to come back to the cell and so on. Okay. But you can read. Yeah. Okay. So this is kind of like the quick summary of it. So is C4 helpful? If C4 is helpful, why rice is not C4? Why many of the plants, your rambutan tree belong to your opa, you know, your kebun pisang that your mak long do not want to let go. They are all C3 plants. Why they are not C4? Why? The question is why? If it is so helpful. Kenapa? Why? Nevertheless, I don't know whether you mentioned here. This evolution has happened at least 60 times in the plant lineage, meaning that it's not only happening in grass, it's happening in other plants as well. Yeah. For example, the salt bush, that's amaran, that's bayam, that's not grass. Okay. So that is the question the scientists all over the world is trying to address now. Why some plants are evolving, some are not evolving. So meaning that it could be more than just simple environmental factor trigger. Just because you live in Mexico, you get C4 photosynthesis. Maybe there is more than that. Right. I don't know, mungkin kawasan tu ada orang berimai yang duduk, then the evolution happens faster. Right. It can happen that. Yeah. Because if only happens in one region, how do you explain it happens in non-desert region? Right. Because not all Mexico is desert. Not all Brazil is desert. Right. Brazil is pretty much like us. But they have this evolution going on as well. Yeah. Right. Okay. Many of my friends that I know they're doing this study actually. So many plants they are in the transition between C3 and C4. They call it intermediates. And much of it is actually grass. Okay. So that's the first part of it. Aku sekejap lagi lah kita buat. Kita baca. Alright, so that's all for the first part of it, the C4 metabolism. Nanti tengah hari we'll continue the final metabolism of it, which is the CAM. Okay, any question? Ada bangkangan? Sila baca nota anda sebab benda ni memang kena baca berkali-kali. You know, like I said before, you should feel privileged because the book I'm using this is the latest book dalam dunia ni. That's why you get all this. You can check on Wikipedia, mungkin tak ada lagi benda ni. Because this is baru. The book was published in July baru ni. Yeah. No. Library tu dah 10 tahun belum tentu ada lagi buku tu. Alright. Okay. So I particularly like this figure here because this is actually the figure that I used. Dulu kononnya aku nak pergi Cambridge. So I did a proposal for my PhD kan. This is the image that I used actually for my Cambridge proposal. Nampak orang katakan bodoh tak dapat masuk Cambridge. Is that really so? I'll tell you lah, because ada some people I know kind of heartbroken sebab tak dapat masuk MIT. You know MIT? Tapi dia engineer lah. Actually kan, tempat-tempat belajar ni memanglah it's important. You need to look at the reputation of the academic institution. But actually depends on engkau, whether engkau tu rajin mencari resourceful ke tak, and so on. So this thing actually I use for my proposal because masa tu the C4 project just started. C4 project just started and I kind of saw something, a way to do it. So Cambridge actually accepted proposal. That actually makes sense. Otherwise dia takkan terima in the first place. So had to go through a series of interview, which is sangat susah. Dia tanya bukan soalan pokok je, soalan planet mana-mana dah dia tanya. I think berapa soalan ni total? Macam 70 kot. Dia call UPM, dia call. Bukan kat sini dulu. Kita ada fakulti lama. They call to the room. Satu je soalan tak dapat jawab. Dia tanya the name of the gene yang buat rubisco. Rubisco ada dua parts, right? So one of the gene tu I kind of misspell it kot. RBCL, rubisco large subunit apa tak. And the other guy from Holland dapat jawab. Sebab tu je dapat. Itu je sebab dia. Lepas tu kena reject. Kau rasa apa? Sakit hati tak? Sebab the place is only one. Sebab sistem kat Cambridge and Oxford, if you get the admission, automatically you get the college, you get the scholarship, you get the tutorship, semua macam-macam. Automatic. Regardless whether your scholarship is coming or not. Dia bagi placement macam tu. Because dia macam, you watch Harry Potter kan? The sense of belonging to your house or to your college is sangat tinggi. Bukan macam kita kat sini, macam kau tak ngaku kau pada serumpun. nobody wants. No. People kat Oxbridge ni, Oxford Cambridge ni, they are very proud from which college they're from. From the Girton College, from the Trinity College, from the King's College. Dia macam Harry Potter tu. Macam kau kan kata, Harry Potter kau ni serumpun. 14. Paling cikai kat sana. I think yang baru-baru lah kot. Dia tak ada lah cikai. Dia kurang real estate. You see all these colleges dekat Oxbridge, kaya tau. Kaya sebab dia ada real estate macam rumah-rumah, ada dia punya ladang buah, ladang pokok, apa benda semua. So they're very rich. Tapi yang college yang baru-baru ni, contohnya college Hazy, Robinson, dia macam dia tak ada sangat benda. Cuma dia ada tempat orang buat teater. Tu satu je dia ada. Tapi they are closer to the modern facility. Contohnya macam Photonic Institute untuk study photon. And this place is actually dekat dengan tempat Sumita. You know, Sumita sebelah tu. Sumita sebelah silen. Kau tak ambil kelas like apa animal science ke? Kau dia tak kerja dah sekarang? Adalah. So Sumita dia pada situ juga, dia duduk kat belakang ni. Sebab dia school of animals. Yeah. So is that a bad thing? Actually kan, because it's been over 10 years now, not going to Cambridge is one of the best thing happen to me. Sebab if I went down to Cambridge ni, sampai sekarang C4 ni masih orang tak jumpa. Satu. Nombor dua, the competition is so hard, I wouldn't be having the experience to explore the science in any way I like. Sebab terlalu fokus nak dapatkan C4 ni. Tak ada ruang untuk berjalan dan bergembira sangat. And the third one is, orang kat sini, you can say that dia agak proud. Okay? Dia memang, British tu dah memang tak friendly sangat. Dia reserved. Lagi tak friendly. So ada banyak kat situ. So disebabkan dapat tu, I got other university. Tapi life is so much colourful. Yeah. And guess what? My finding is even more better than the original C4 rice tu. So just because dia tak jadi mula-mula doesn't mean that that's the end of it. Memanglah ambil masa lama, lebih 10 tahun. So apa dia aku dapat tu? Oh macam-macam lah. Takkan nak cerita kat kau. Tu aku buat buku. No, just don't feel bad lah, because I got my friend nak going to MIT tapi tak dapat engineering lah, tapi you just don't feel bad about it? Kan? Just one door closes, the whole garage will open for you. Okay, all right. So, any question? All good? All good? All good? Okay, so I'll see you in the afternoon, okay? Let's finish this off. I think kejap lagi kita discuss very quickly petang nanti. So please datang. We need to because I won't be around for 2 weeks, the final week, 13 week 14. So we might need to ada kelas ganti 2 to finish this off, and then untuk buat 2 session lab sebab nak demonstrasi alat. And then you are left without me for about 3 weeks. Betul tak Izan? Bye. Alright, okay. See you.